Oh, that's weird. So this is my first time actually using this computer here, so that's why I'm freaked out a little. It's giving me this really funny display, but I'm gonna go with it. Anyways, I'm Alana, but don't call me that unless I tell you to, Dr. Ehrlich. And welcome to 408. Who should be in 408? Who shouldn't be in 408? Okay, you guys are about 400 level. You should know what you're doing, right? Yeah, I thought so. Um, so let's go through a little bit of business. It's technically 10:59 according to my computer, but we're going to go with it. So some business. Syllabus is on Blackboard. Most of you, actually I checked, 35 of you have looked at it. There's 66 of you officially, so that's a lot of people who haven't looked at it. So please look at it. Actually, who was it? Dr. Nelson. Who's had Dr. Nelson? He, I believe, once tracked this who was looking at his syllabi and realized that there was like a huge percentage of the class who never even opened it on Blackboard. so now I'm tracking. So I'll know who you are when you ask me silly questions like, when does recitation start? Because actually, I'm going to say it a few times today, and it's on the syllabus. Anyways, so save it to your computer if you don't like paper. Like I hate paper, so I get that. Print it if you love paper. I know people who only read things on paper, so that works. Look at it. Look at it. Okay, there's a pretest. It will be available today. It will be available today at some point, and is due by midnight pretty much, 11:59 p.m. In 59 seconds on Friday. And it's just testing you on stuff you should know. You should not have to even think. It's like, what is protein? Like, if you don't know what protein is and you're here, see me. So that is worth 10 points of your total grade. It's an easy 10 points. I'd like you to just do it and not spend your life looking up the answers, but I can't stop you from cheating. I'm just saying. Please don't. There's a weekly quiz, and that starts on Friday. I'm sorry, it's Labor Day weekend. So after you go to the state fair, who's going to the state fair? Yeah, I'll be there. I'll be there. After you go to the state fair, or you can go to the state fair on Monday, because that's the last day. So you can spend the weekend doing your quiz, and then go to the state fair. The quiz, a lot of people hate the fact that I give a quiz every weekend. My apologies in advance, but it will push you to prepare for the next week because I'm not quizzing you on what I taught you. I'm forcing you to teach yourself what you're going to learn next week. And if you're angry about that, then leave because there's like 12 people on my wait list who will be really happy to see you go. So you have to be okay with the fact that I'm going to force you to teach yourself, and then I will be here to help explain and clarify. A lot of students have actually said they like this because it means that they have to think about things before they come to class, and then they're not furiously scribbling everything down. So this right here, this girl right here, it's only 11:02, so she's good. But by 11:05, that door will be locked on a normal day. Today I'm going to be nice. Today I'm going to be nice. Because see how distracting this is? We're all watching who's walking in late. So just so you guys know, it was okay in that other room for those who had 409 with me because you can sneak in through the back. But here I'm totally going to lock the door at 11:05 every day because you guys don't need to be distracted, right? Anyways. Now that I'm done being distracted, quiz. Really great opportunity to kind of, think about what we're going to be learning the next week. And you come in and you already know what we're learning and you can think about it and you can ask thoughtful questions. It gives you so much more time. And the quiz, it's not just recall, it's hopefully questions that are going to make you think about things because I really want to teach you guys how to think. Ideally, you already know how to think, but think at even a higher level than you already do. Okay, office hours, they're 9:30 to 10:30, so right before class, Monday, Wednesday, Friday. That might suck for some people. It works well for me, but that's why I write or by appointment. So always email me. If you're not around between 9:30 and 10:30, I get that. Just email me and we'll find a time. I'm always here to help you. If you come to me and ask me for help, I will make sure you do well. But if you don't come to me until the last week of class and you're failing, I don't care if you fail. Okay? You guys are adults. You need help, ask for help, and I will be here for you. That leaves me to ask for help. Please ask for help. This is not easy. And especially the way I teach it, at times it cannot be easy because I harp on thinking and big picture. And I'm not going to give you a study guide which tells you what to memorize. So, see, distracting. Definitely ask for help. Okay, recitation starts 9:10. So that gives you a little while. Are we excited? We have some time to ease into the semester. You know, it's been summertime. We've been relaxing. Who is relaxing this summer? Okay, that's good. Who is working their butt off? Yeah, I know. Isn't it unfortunate? It's just the beginning. So recitation starts on the 10th. And papers, it will be in the format mostly of you read a paper at home and there are discussion questions and you'll work with your groups to go through the papers and discuss things. And then at the end of recitation, you'll take a quiz. That quiz will be based on what you did during recitation. So there's really no way to do badly if you prepare and you participate. If you don't prepare and you don't participate, you will do badly. And well, that's your problem. Okay, quiz at the end of each recitation. Okay, all lectures are recorded and available online. There will be on Blackboard just a link. This year I have a YouTube channel. Is that exciting? So I have a YouTube channel, and so the whole world, I guess, could see it. So there'll be a link, and you guys can watch it. And a lot of people, I think, have found that to be helpful in the past because sometimes I just ramble on and on and on, and you can go back and listen to it again. Also, I say really dumb things sometimes. So you can go back and relive that if you're feeling dumb. Be like, well, Dr. Ehrlich was really dumb in class yesterday. And that was really cool. Okay. So, and the wait list. So there's, like I said, like 12 or 13 people on the wait list. I'm very sorry that we don't have enough people to teach this class. As you can see, this is a pretty full class. So I will see who shouldn't be here. Yes?
In the things online, are we going to be able to see the PowerPoint?
Yes,
Oh,
yes.
great.
Yeah, so the recording is PowerPoint. Oh, it'll be interesting, actually. We'll see how it looks because it's like this really funny display, but it is a PowerPoint in my voice. Yeah. Yeah, so those who are on the wait list, please come up after class. I'll check you off because you get credit in my brain for being here, even though you're not in the class, and you will move up on the wait list, and those who did not show up today will move down on the wait list. But the way I've organized it is those who are graduating this fall have priority. Those who are graduating in 2 years or are not students are lower on the list. You know how it goes. Those who are graduating this fall, you should have gotten into this class, or I don't know what happened. I think that's that. Oh, look, we can change the slide. I guess towards the end of class, I'll take attendance so we can try to figure out who's not going to be coming. Okay, so you're thinking, if all your lectures are recorded and online, why should I bother coming to class? True, you don't have to come. You could probably do fine. That's great because then we can have these intimate conversations if no one comes. But the reason you should come, aside from the song and dance, is I have all these great extra credit opportunities that you'll only know about if you're in class. So like when some brilliant student raises their hand and stumps me, I put it out as an extra credit for you guys. And you have 24 hours to come up with the answer. And the gals, whoever who come up with the answer and email it to me within the next 24 hours get a point. I do that. How many times have I done that? People who've had me before. A lot, right? You can probably get like 10 extra points or more if you do that. So it's worth showing up. And then sometimes you might have a question which you might want to ask in class. And also, obviously, other people's questions are usually helpful. So it's useful to come. But then again, if it's 11:05 and you're not here, I'm going to close this. Since it's 11:05, if it's 11:05 and you're not here and you get locked out, it's not the end of the world because you can listen. You can listen online. Extra credit. So people always flood me with requests for extra credit at the end of the semester if they're not doing well. And not doing well, there's a range. There's a I need an A not doing well, and there's the I need a C not doing well. people have all sorts of. I usually think if you are worried about getting an A-, I don't want to see your face. But, we all have our thing. So lots of extra credit points. There's the stump the teacher points, like I just said. If I can't answer your question and you find the answer, points. You always have the option to see me about presenting a relevant, so something we're talking about in class, pre-approved research article in class. It might be 10 minutes. You get up here with a PowerPoint of like 1 or 2 slides and you say, hey, these people, they did great things. Let me tell you about it. Here's a few figures. And it's useful because you get to learn a little bit about how to read primary research articles. I would help you prepare for that a little bit. Not like I won't go through the paper and tell you what to do, but I would at least, look at your final product before you get up in front of class. Another option is you can create a multiple choice question for an exam. So usually before an exam, I'll put out a call like the week before for multiple choice questions. If you can create a good one, and I'll put up criteria online. Like you can't, they can't be all of the above questions. Only I can do that. Then you can get not only the points for getting that question right on the exam, but also some extra credit points. And then usually at the end of each exam, I have a 10-point extra credit. So there's a lot of chances for extra credit, but get on it now. It's the first day of class. Get on it now. Don't wait until you're failing. Not that anyone fails this class. Everyone does really well. Right? Positive thinking. Okay. Cell phones on silent. That goes without saying. Please don't text or look up cool things on Facebook or whatever you do while you're sitting here because it's distracting. I get distracted so easily. Like a bird flies by, I'll be like. So I don't want to see you guys doing crazy things with your technology. Otherwise, I'll say, can I have your technology? Because my kids need things to play with at home. My son, he's 2, and he loves buttons. So like an iPad or a laptop. Buttons. You guys, come on. 11 a.m. I was hoping to have a real live audience. Okay. You can pick up. I won't give it to my 2-year-old. Okay. Don't be late. I heard said that already. Let me know in advance if you're going to be late. If you are coming from a job interview or something that's legit or a doctor's appointment, just email me the day before and say, hey, I'm going to be late. Don't lock me out, please, please, please. And I will actually leave the door open for you, and then I'll give evil eyes to the other people who sneak in. Okay, so things you should know from 201 and 309, things that will be on the pretest. These are major topics. If you've been through 409, you probably know most of this also, but that's not a prerequisite for this class. So I think it should be. Nucleic acid structure, DNA replication, protein synthesis, genetic code. These are the basics. You've learned them how many times? You've learned them at least twice by now. And so this is just me making sure that you know this stuff because I really would rather you not struggle through a class. Has anyone in this class not taken 309, which would be genetics? No. Okay, good. That means the registrar was doing what they should do. Okay. So this is if you kind of think about these things a little and then take your pretest, then you should be good. I will not spend much time harping on this. I'll spend a little bit of today and a little bit of on Friday kind of going over review kind of stuff, and then we're diving right in because I don't like to talk about what amino acids are because I feel like I get lucky. I get to teach the upper level classes, so I don't want to teach the basic stuff. Okay. Hey, professor. Any advice for how to do well in Bio 408 and 409? This applies for both. Well, I like this cartoon. We'll have lunch here, and then we'll keep looking for whales. So this is a way that a lot of the time students look at science. They just look at the little parts, and they don't think about the big picture. And I've had people come see me in my office in a panic after the first week or two. I'm looking at one of those people. And I said to them, just every time I tell you something, ask yourself, how does this fit into the big picture? Because I won't tell you about it if it doesn't fit into the big picture. And if you're having a hard time figuring out how it fits into the big picture, ask your friends, ask me. But when you start to think about things as, This is a cell, and the cell needs to live and function and survive. And all these little details that you think I'm telling you about, they all fit into that goal of surviving and doing a job of some sorts, right? Kidney cells do kidney things. Heart cells do heart things, right? Right? Big picture. That's how you do well in this class. If you start memorizing details, you will fail. I'll tell you now. Memorize details, bad. Think big picture, good. Okay? That is how you do well in this class. And just stop yourself all the time and be like, okay, why am I memorizing this? Why is this detail so important? Probably isn't, and probably you just need to think about the big picture. Okay, so what is cell biology and what is molecular biology? I'm really interested about this actually because cell biology and molecular biology seem to have very different populations of students who take them. Partially, I think, because some are required, some people have to take both. But those who don't have to take both often choose cell biology. And really interested in why that is. How many of you guys think cell biology distracting? How many people think cell biology is easier than molecular biology? Who can tell me? I've been talking at you guys for too long. I'm sorry. Who can tell me? I hate when I don't ask people to talk in class. Does anyone want to add anything? No? Okay. So, who can tell me the difference between cell and molecular biology? Anyone? Yeah.
Cell biology is outside of the cell, and molecular is inside of the cell.
Well, pretty much what you meant to say is nucleus, right? So I usually talk about cell biology as everything happening outside the nucleus. Now, Dr. Megan May, those who've had her, would kill me if she heard me say this because she works on bacteria, and do bacteria have a nucleus? Right, so it's like a whole large amount of the Earth's population that I'm ignoring when I say that. So my apologies to Dr. May and her students who are in this class. Don't tell her I said that. But yeah, so I see everything that is DNA related, we'll say it that way, like anything that has to do with DNA replication or anything that has to do with gene expression, and regulation of gene expression, that's molecular biology. And cell biology is everything going on outside the sphere of the DNA. Like everything from translation onwards, so translation of proteins, and they need to go where they need to go and do what they need to do, and the communications between cells. But in reality, there should be one class. Because in reality, it's cell and molecular biology. It's one thing. Cell biology is the nucleus in the cell? Yes. Right? Nucleus in the cell. Who thinks the nucleus is out of the cell? Okay, good. You're all awake. So I think that it should be one class, and it should be a year long, and you should have to spend quality time with me for an hour every other day for a year. Would you love it? I'll ask you again at the end of the semester. So I put this picture up because this is a immunofluorescence picture. I love immunofluorescence. So this is immunofluorescence where the DNA is stained with DAPI, so it's blue. Sorry, laser. Blue. So there's 1, 2, 3 cells there. I'll show you pictures like this a lot, and you usually can orient your cells by looking for the blue. Because DNA is in the nucleus. Usually there's 1 nucleus per cell, right? What happens when there's more than 1 nucleus per cell? Bad. Cancer? Okay, bad. So you've got mitotracker red, so that's the mitochondria. And then you've got phalloidin, which is cytoskeletal actin. So that's your cell. And we're going to say cell biology is everything happening out here. So I guess we'll talk a tad about mitochondria because we'll talk about import of proteins into the mitochondria. We'll talk a whole lot about the cytoskeleton and actin and microtubules and how that's all organized and how the cell moves. Inside the nucleus, you'll have to see me in the spring in 409 And we'll talk about all sorts of great things with DNA and histones and fun things like that. Okay, so that's what each of those things are. I love immunofluorescence. We just got a new microscope in this department. It's really exciting. Excited. Any questions so far about what I'm talking about? It's not really that deep yet. Okay, good. Okay, so this is my favorite exercise for the first day of class. Let me get my cheat sheet out here. Okay, the cell. Let me get some chalk. You guys are going to tell me about cell biology. Cell. Okay, so when I was in graduate school, who's going to go to graduate school here? Some sort of graduate school. Who wants to get their PhD? So, when you go get your PhD, they make you do horrible things like oral exams, which, by the way, I think everyone should have to learn how to do because you get over the nervousness. But anyways, so I had two. It's like at the end of my first year and then at the end of my second year, I had oral exams where like you're sitting in a conference room by yourself with like 5 brilliant faculty, because faculty are always so much smarter than you. Not really. And they're asking you questions. And I get up there and they're like, okay, draw a cell, draw everything in it, and tell me what each thing does and how a gene gets from DNA all the way through to out of the cell. Who can do that? You guys can all do that together. So we're going to do it together. And by the end of this class, you'll really be able to do it. Because I think it's important, because that's the big picture, right? Okay, so let's talk. So I just drew a circle representing a cell. So what was that circle? What is that? Okay, so that's the membrane, right? Plasma membrane, cell membrane. Okay, so what's inside that? Cytoplasm, okay. So that's just... We're going eukaryotic, thank you very much. But let's point out the differences here and there, yeah. Right, because then we'd have to talk about all sorts of things if we were going prokaryotic, but we're not going to go there because I work on mammalian cells, so I deserve my bias. Okay. Plasma membrane. Okay, so let's raise hands. So what else is in the cell? Yes?
You have a smooth and rough ER.
Okay, smooth and rough ER. So where would that be? What's that attached to? Around the nucleus. Okay, so we're going to have a nucleus. Or if you were not a eukaryotic cell, no nucleus. Is there a smooth and rough ER in a bacterial cell?
No.
Ooh, I see yeses and nos. Let's think about that. Okay, so who's taking 315? Okay, you guys will know. Are there membrane-bound organelles in bacteria? Okay, there you go. Is a smooth and rough ER membrane-bound? Yeah. Okay, so let's do smooth and rough ER. Looks kind of like that. Okay. Why do you think it's so close to the nucleus? Why? Well, before I say that, what is rough ER? Is it good for exfoliation? Yes.
Smooth ER with ribosome.
Right, so it's just smooth ER pretty much, but with ribosomes. And what are those ribosomes doing? Right, they're making protein. They're importing they're basically translating proteins right into the ER, right? So we'll do some. There are ribosomes. Do you think there's one ribosome? No. Okay, just making sure. When I came out of high school, I think like they teach you the cell, the ribosome. It's like there's this one like master protein making machinery. Okay. There we go. Making proteins into the rough ER. And we'll do that. What's the smooth ER? What does that do? Lipid synthesis. That's one thing. Yep. And then there's something about calcium storage and release. Okay. Anything else? So we talked about the nucleus. What's in the nucleus if you're a eukaryotic cell? Chromosomes. Okay, DNA in the form of chromosomes. Exactly. So what makes DNA chromosomes? Like, yeah, histones, right. So the DNA is wrapped really tightly around histones, chromosomes. Okay, so I'm going to draw the DNA as condensed chromosomes. Just two. Right? Is there that many? No. What phase of the cell cycle do you think I'm putting the cell in? Huh? When is chromosome condensation happen? Mitosis, yeah, prophase. So the beginning of mitosis, right? So remember the cell cycle: S phase, M phase, G1, G2, right? So we're doing a cell that's getting ready to enter mitosis, right? Because you got to condense your chromatin, because when you split things apart, if it's not condensed, you'll tear up your DNA. That's bad, right? Right? DNA, stay together, breakage, bad. Okay. So we've got our condensed chromosomes and our histones are all in there, right? Okay, so let's go back. So we've got our rough ER near the nucleus. Oh, so distracting. So distracting. Come on. Okay. I'm going to have so much fun locking that door. Okay. So why is the rough ER near the nucleus? Why would it be there? Why isn't it near the plasma membrane? Anyone ever think about it?
Like, remember that mRNA is leaving, it will just like translate protein.
Right. So the mRNA is leaving the nucleus, and then a lot of mRNAs need to be translated into the ER because they're going into the secretory pathway. So it's really convenient, right, to have it right there, right? It wouldn't be very efficient if the mRNA needed to travel across the cell to be made, to be translated. Right. I saw another hand up somewhere. Anyone want to add to it? I think that's a good answer. Okay. What else is in the cell? Is that it? Okay.
The Golgi.
Golgi. And so the Golgi will often kind of be like that, kind of like coming off of the ER. What does a Golgi do? Right, so it's kind of involved in packaging. Well, that packaging actually starts in the ER, and then it goes to the Golgi, and then things that are going to be secreted out of the cell, or things that are going to live on the plasma membrane, like membrane proteins, they are packaged and they make their way from the ER to the Golgi to wherever they need to go, in terms of secretion, membrane proteins, and lysosome. But what's happening in the Golgi? Is it just like the FedEx Kinkos? Actually, now it's just FedEx Office or something now, right? No more Kinkos. Goodbye, Kinkos. Who uses Kinkos anymore? Do you guys use Kinkos? Okay, well, there's a lot of modifications that need to happen to proteins, right? So in the ER, you're having translation of proteins that are going in certain directions, but a lot of modifications need to happen, and some happen in the ER and some happen in the Golgi, and we'll go into that even further, and then they get sent to where they need to go. What do I mean by modifications? What's a modification? What am I talking about modifying? Proteins, right. Proteins need to be modified before they go to wherever their job is, right? And so what kind of modifications are you thinking about in terms of ER and Golgi? Yes?
Folding and structure changes.
So folding and structure changes in ER, right? And Golgi. Do you like how I'm making you learn the whole class on the first day? Yeah. Phosphorylation we will learn about in signaling, and that actually happens in the cytoplasm or the nucleus, and that more has to do with modification of the protein in terms of its function in the cell. So like a signaling protein will be turned on or turned off based on its phosphorylation state. That is a post-translational modification. You're absolutely right. Just not one that happens in the Golgi. Yeah. Yes.
Proteins need to be packaged, right?
So they need to be packaged, but so one thing I'm looking for is, let's say it needs to go to the membrane. Membrane proteins tend to be highly, highly what? The part that's in the membrane. Yep. Yep, you guys are going, okay, I'm pushing you guys really hard here because you're doing so well. Hydrophilic, hydrophobic, depending on where they are in the membrane and in the outside and the inside. But what I'm looking for is glycosylated, right? Adding sugars, right? So membrane proteins and secreted proteins tend to be very glycosylated, and that happens in the Golgi. That's actually one of the reasons it's been really hard to make an HIV vaccine that's targeting the envelope protein, because it's highly glycosylated, and it's really hard to make an immune response to highly glycosylated proteins. Anyways, okay, what else? So we've got Golgi. What else? Yes? Mitochondria. Okay. And we'll do like little bacteria in the cell. Okay, so what's mitochondria? What are two major functions of mitochondria? I heard the word ATP. ATP, so energy, electron transport, all those kind of things, right? Which I'm going to completely ignore in this class. I'm sorry. What other major thing happens with the mitochondria, which you probably don't know about, but I think is really important? Yes? Protein synthesis. Our protein synthesis in the mitochondria. Well, yeah. So there are, mitochondrial DNA in that. Yep. But more important than that, what happens if a cell is doing really badly, like the DNA is damaged? Then yes.
It can trigger
Apoptosis, absolutely. And that happens by poking, there are many ways it happens, but one of the ways, which involves the mitochondria, is by poking holes in the outer membrane of the mitochondria and releasing cytochrome C, and that starts the whole cascade of caspases and all that, and chopping up everything in the cell, and apoptosis, right? Okay, so what are we missing? Okay, there's some things. Yep. Lysosome. What does the lysosome do? Lysosome. Yes. Digest stuff. It's full of a lot of proteases and just enzymes that degrade things. So there's two ways of degrading things in the cell. Well, first of all, why would you want to degrade anything in the cell? Yes?
They're toxic.
So things that, like misfolded proteins, things that are toxic, things that shouldn't be around. Yep.
Ineffective or damaged proteins?
Ineffective or damaged proteins. Yep.
It's almost the same as eating.
Same as eating. So there that we go into autophagy with that. Yep. Well, another thing is sometimes you want to turn something off, and we'll learn about that a little bit more when we talk about signaling. But if you have some process happening in the cell, usually it's started by proteins talking to each other through phosphorylation and going to nucleus and activating gene expression, and that's how things happen, right? If you want to turn something off, the best thing to do is to degrade your messenger, right? Now, usually that's not by the lysosome, but it is actually frequently by the lysosome when you're talking about receptors, right? You have a receptor on the cell surface, it's signaling, it's telling another cell something, or whatever or it's receiving messages, I should say, from another cell, you want to turn that off, endocytose that receptor and send it to the lysosome, right? What if you want to bring in certain kinds of lipids from outside and you want to scavenge? You can endocytose your receptor, bound to your lipid, bring it to the lysosome, right? So, things to think about. Another way to degrade things in the cell. Yep. Not peroxisome, but peroxisome is peroxide production, but it's not an organelle, but it's a large machine that I'm in love with. Proteasome. Okay, proteasome, ubiquitin proteasome. It's a big one. You'll hear it a lot. Okay, so lysosome, degradation of things. Okay, so what else? So we've got that. So you brought up peroxisome. That's another thing that we didn't mention, right? So that's one that I don't know too much about because I've never worked on it personally, and we won't talk too much about it, but it has a lot of enzymes and it makes peroxides. Okay, so what else did we forget? Anything else? Anything else? Ah! Yes.
Are centrioles in this one or?
So centrioles are in the nucleus, and what are centrioles? Does anyone? Now we would probably focus a little bit. Well, we'll talk a little bit about it. A little bit. Has to do with mitosis and separating your chromosomes. Yep. Cytoskeleton, right? So cytoskeleton. So that's a really important thing because what does a cytoskeleton do? Shape, yep. Movement. And so when we talk about the cytoskeleton, one of the big things that I think is interesting is, okay, shape's important and resisting stress and tension. And like think about that. Think about like what a white blood cell or a red blood cell has to go through, right? Going through like blood vessels. Think about like the pressure on it, that blood and rushing around corners. And it's important actually to have a serious structure, right? Otherwise you're going to be popping on, leaking all over the place. Okay. So cytoskeleton. What is a cytoskeleton? So that's mostly made of what proteins? You guys have any idea? We'll learn more about it. Yep. Fibrin. Oh, maybe not what I was looking for. I'm thinking that's like extracellular matrix, right? Is it? Yep. Actin. Yes. Okay. So don't take me out of the cell because I get confused out of the cell. I'm just saying. You know, like you guys, everyone has their specialty. Don't take me out of the cell. Don't put me in a plant. No plants. So I think that's extracellular matrix. But actin. Okay, so you've got actin filaments that are all over there. And those things are really dynamic, and they actually are constantly, so they're made up of these subunits, and they're constantly building and shrinking, and that's how the cell moves, through signaling that results in building and shrinking in the right parts of the cell, right? So if you want to move in this direction, you're going to end up kind of like getting rid of some actin over here, sending it over here, and building in this direction. And then you get these, filopodia and movement. What else? Actin and what? I know you want to say myosin, but that's not what we're talking about. Yep. Kinesin. So that's a motor protein. That's a motor protein that's involved in moving things on what? I'm pushing you guys hard for the first day, aren't I? Tubulin. Okay, so that's one of the others. So actin and tubulin, those are my big cytoskeletal proteins that we're going to talk about. And so we've talked about motor proteins on tubulin, and you can think of tubulin as little train tracks in the cell. And they're everywhere, all over the place. Maybe not going through the nucleus, but they're all over the place. And They help to move things around. Have you ever wondered how things get from one place to another in the cell? Do you think they just kind of diffuse about and randomly get places? A lot of the time things get moved around, and we'll look at a movie showing this later on. And so one thing is, like, we've got all this movement of organelles, movement of Golgi vesicles. So a lot of this is on tubulin and motor proteins kind of moving things around. Also tubulin, microtubules involved in a major phase of the cell cycle called mitosis, where we have to segregate chromosomes, right? So there's the mitotic spindle that is made of microtubules. Okay, what else? I think that might be it. So that is the whole semester. Thank you for coming. I'll see you guys in December for the final. You guys teach it all to yourself. It would be fun, huh, if you guys can just do that and come back at the end of the semester. No, I'm just kidding. Okay, so that's it. That was a fun exercise. Okay, let's move on. Thank you for all who participated. Okay, so this is someone's foreign and domestic DNA repair shop. It says, you're lucky no one was injured. Your base pairs are out of alignment, and that has your reading frame all messed up. What does that mean? Your base pairs are out of alignment and has your reading frame all messed up. So we're just going to talk for a minute about things that you should know. And we'll start now, and we'll finish on Friday with that. So when we talk about base pairs, what am I referring to? Base pairs. Someone raise your hands. Show it. Yeah. Huh? Nucleotides. Specifically, what form of nucleotide? Base pairs. Mostly, I'm talking mostly about DNA when I think about base pairs. But yeah, nucleotides. We'll go with nucleotides. Reading frame. What's reading frame? Yes.
Where to start reading. If you have a gene, DNA, you have a place to start reading it.
So there's a start and a stop. And reading, what are we talking about reading? Yes.
Reading
Codons, right. So if you've got your gene and you've got your start and your stop, TAA, that's a stop codon, right? I don't know. And then every 3, right, are your codons, and those are your, you translate them to make your protein. And the frame, the reading frame, means you started here and every 3 here. Another reading frame would be if you started here. Or another reading frame would be if you started here, right? So if your base pairs are all out of alignment, your reading frame might get messed up. If your reading frame is messed up, what's the result in your protein?
They don't.
Much more than mutation, right? It's a completely different protein if you have a frame shift, right? Often it won't work. Sometimes you'll have a stop codon created where it shouldn't be, and sometimes it's just a bunch of gibberish and it's not a functional protein. So when we talk about, you talk about insertions and deletions, that's the major issue is you have a frame shift, right? Okay. So, truths and misconceptions. So here, this slide here is my nemesis, okay? And I'll tell you why. Last semester, I had a horrible discovery, and we're just gonna test this one right now. How many people believe that before transcription, you have DNA replication? Feel free to raise your hand. I promise I won't judge you. About 60% of my 409 class thought that. So do you need to replicate your DNA before you can do transcription? Yes or no? Okay, great. I've been actually telling all the other faculty to hammer that in. So the problem is when you're taught this, you're taught all the time DNA, RNA, protein, replication, transcription, translation. And people start to think that you need to replicate your DNA and then you turn it into RNA and then you translate it into protein. That is not the case, right? So think about it. Replication, when does that happen? when the cell's dividing, right? So in S phase of the cell cycle, you replicate your whole genome because you're going to turn it into 2 cells, right? You're going to divide into 2 cells, and each cell needs a genome. That's replication. Why they put it here, I understand DNA has the information, which is then to RNA, then protein. I understand that, but this is actually the source of so much misconception. It makes me crazy. So I want everyone to really understand that replication is cell cycle, right? And RNA into protein, right? That's really gene expression. Okay? So yeah, you have to find your gene that you want to express, transcribe it into RNA, and then the RNA process goes out of the nucleus, meets up with the ribosome, and it's translated. That happens, absolutely. But no, you do not need to replicate your genome every time you need to express one gene. Okay? Is anyone wondering what the hell I'm talking about? Is anyone like, why is she talking still? It's 45 minutes and she's still talking. Okay, good. So there you go. So you got your DNA, which does hold all of our brilliant information. And then when you want to express a gene, it does need to be transcribed into RNA, and that will be translated into protein. Okay. So what is DNA and RNA made out of? Huh? Nucleotides? Someone? Okay. What is protein made out of? Okay, that's another one I just need to make sure we're all clear on, because I've heard of people graduating thinking that nucleotides make proteins. Not good. You're not allowed to graduate with that knowledge, without that knowledge. Okay. DNA replication. So when does DNA replication happen? S phase of the cell cycle. Why does it happen? Good, thank you. I love it when people shout out wrong answers. DNA replication does not happen to express a gene. It never happens to express a gene. It always happens. You know, it's important to tell me the wrong answers, because that means probably 20 other people are sitting here thinking the same thing. DNA replication only happens when you're about to divide. The cell's about to divide. You never replicate your DNA. You never make a copy of your DNA before you express a gene. Never, right? It's not very efficient. Let's say you are eating donuts and you have a rise in your blood sugar and you need to make more insulin. Are you going to replicate your whole genome just to make more insulin? No, it doesn't really make sense, because the genome's huge, right? There's a lot of genes in there, right? Okay. So why we replicate our DNA? Because we need to make daughter cells, right? And everyone needs a genome. Okay. Reading the entire genome. You know what? I'm going to stop right here because I want to spend a little time taking attendance.